Hi, I'm Ryan Morrie, a postdoctoral fellow in David Julius's lab at UCSF. And today, I want you to think about the most recent stomachache you've had. Maybe you took some Pepto-Bismol and then went away. But why were your guts bothering you in the first place? Did you eat something pungent and spicy? More importantly, how did your brain know that something was wrong in your intestines? Most research on sensory signaling, or the biology of how we perceive the world around us, has focused on organs such as your nose, eyes, or skin. But your gut's also a specialized sensory organ. In fact, it's the largest surface area of your body exposed to the outside world. As you can imagine, sensing the types of nutrients or toxins present in the things we eat is essential for survival. To accomplish this, your intestines use many of the same types of cells and molecules as your nose, tongue, and skin to smell and taste the food you ingest, or sense touch during stretching of the gut, like when you're too full. The gut sensation I'm most interested in exploring is pain. While gut pain normally helps to keep us healthy by telling us when we've eaten something we shouldn't, pathological conditions can develop, in which normally innocuous sensory signaling erroneously leads to the sensation of pain. To treat patients with irritable bowel syndrome or Crohn's disease that are suffering from these types of symptoms, we first need to understand the fundamental biology of how these pain signals are produced. With my background in basic neuroscience, I believe I can provide a fresh approach to understanding and eventually treating these understudied but prevalent medical conditions by utilizing cutting-edge techniques to identify the cells and molecules responsible for pain sensation in the gut in both health and disease. So where does sensory signaling in the gut begin? Recent work from my lab, as well as many others, has found that not all the cells that line your intestines are the same. While most of these cells are there to act as a barrier and help you absorb nutrients from your food, the main jobs of the intestine, there is a rare population, I'm talking less than 1% of all the cells that line your gut, that directly respond to chemicals and mechanical forces. Some of these rare intestinal sensory cells have receptors for noxious chemicals, such as smelly byproducts from your gut bacteria, or the pungent molecules in wasabi and garlic. Upon stimulation by these harmful gut chemicals, it is thought that these cells release hormones or other signaling molecules to induce a painful sensation. However, whether activation of these cells actually induces pain, and how those signals are then relayed onto the brain so you can perceive it, is unknown. To begin to answer some of these questions and identify targets for pharmaceuticals that could be used to treat intestinal pain, I'm performing a variety of experiments, some of which I'll outline now. In one set of experiments, I'm looking to determine whether the rare intestinal sensory cells I'm interested in are necessary and sufficient for pain perception in the gut. I'm collaborating with a pediatric gastroenterologist at UCSF on experiments that allow us to quantitate pain perception in mice by measuring the contractions of their abdominal muscles, whose tension is a reliable indicator of intestinal pain state. I have created novel, genetically engineered mouse strains that allow me to specifically activate or inhibit the rare intestinal sensory cells I'm interested in, while simultaneously being able to measure the animal's pain state. I have validated that these mouse models only affect the cells I want to study, and preliminary results from our muscle tension measuring experiments indicate that these cells are indeed important contributors to intestinal pain. Now, while these rare intestinal sensory cells may underlie the origin of pain signaling in the gut, they're not in direct communication with your brain. Rather, a network of nerve cells relays sensory information from your intestines to your brain. My next set of experiments seeks to determine which of these nerve cells is relaying pain information so that we could begin to target them and shut them off in pathological conditions. To do this, I'm taking advantage of special properties of the rabies virus. Animals get so sick from rabies because it travels throughout their nervous systems via points of nerve cell communication. Scientists have created synthetic versions of this virus that cause a colorful marker to be expressed only in infected cells, allowing one to actually see these communication pathways within the nervous system. To identify which cells are relaying pain information from the gut to the brain, I'm infecting the specialized sensory cells I'm interested in with this modified rabies virus. Only the cells that are in communication with the intestinal sensory cells will light up with the rabies color tracer, allowing me to actually visualize the pain pathway from the gut to the brain. Finally, a molecular characterization of these intestinal pain cells and their communication methods is required to understand how we can manipulate this signaling when it goes awry in disease. To identify ways to control intestinal pain cells, I'm conducting a pharmacological screen for inhibitors of their activity. I can grow these rare intestinal sensory cells outside of an animal in a dish filled with a 3D jelly in what are termed intestinal organoids. I'm making these intestinal organoids from genetically engineered mice that allow me to express colorful markers or other molecules only in the cells I want to study. I then perform electrophysiological experiments in which I take a tiny glass electrode, place it onto a cell, and record the minute electrical signals it produces upon activation by harmful gut chemicals. I then apply a variety of different compounds to these cells to see which ones prevent this electrical activity, and hence pain signaling, from taking place. Whichever molecules are able to do this can then be used as the basis for pharmaceuticals to treat chronic intestinal pain. I hope you now have a gut feeling for how intestinal pain is sensed. My research will seek to identify how signals are relayed from the specialized sensory cells in the gut to the brain to enable pain perception. While this work specifically focuses on the routes and mechanisms of information relay in this pathway, I'm excited to be collaborating with an international team of researchers that's working to tackle the questions around gut pain from multiple different angles. Our hope is that the intestinal pain pathways will be mapped out in the next maybe 5 to 10 years. If this is accomplished, we can then find molecular signatures for the most important neurons or cell types we identify to target them for therapeutic purposes. Importantly, many of the assays that I'm currently developing to assess the fundamental biology of the system can then be used to look at the efficacy of the new pharmacology we hope to develop to treat intestinal pain.